Hello all. I appreciate the invitation from conference organizing team, ABB 2023. I am Professor Nilesh Kumar Sharma. So I would like to present today a very interesting topic, that is how the oncometabolites uh, may cure or may support cancer progression and the drug resistance, that is in the context of chemotherapy drug responsiveness. Okay. To address this particular topic, I would like to go to a brief introduction about the institution or the university where I am working. I represent Dr. D.Y. Patil Vidyapeeth Pune, one of the top 50 uh, Indian university. And I am associated with Cancer and Translational Research Lab. Okay. And this particular, and this is the laboratory where I work, and this is the institution. Now, I would like to go for that is, the first is the introduction, or what are the gaps there? If you look at in the field of cancer biology, the highly promising discoveries are being made, and that is also one of the hallmark in tumor biology, that is metabolic heterogeneity, right, is emerging. We know the facts. Also, the metabolic reprogramming is known to help cancer cells to adapt to various drug resistance. In the same line, the polyamines as a part of chemical or the metabolites, specifically acetylated polyamines, are explored at cellular and cancer patients level, whether how the acetylated polyamines are going to contribute for the cancer, the resistance, and also the cancer growth and proliferation. Also, extending the whole idea of oncometabolites in the form of like acetylated polyamine, the concept of mimetic of metabolites as a part of combinatorial anti-cancer therapies are also being explored. Okay. As I said, that oncometabolites, it makes sense because the metabolic reprogramming or metabolic adaptations by the cancer cell in response to the drugs or any other environmental stress, it's quite amazing. And these oncometabolites are also explored in various perspectives, like whether it is, you can say, uh, tumor biomarkers, right? Growth proliferation, also one of the interesting epigenetic or metabolic axis, how the metabolic pathways and epigenetic is connected. That is also one of the example of oncometabolites is connected. Drug resistance. And the most important, the story for this presentation is one of the class of oncometabolites as acetylated polyamines we explored as mimetics of these metabolites, okay? And that is in the perspective of combinatorial anti-cancer therapies. No, the methodology is not so new. We know the facts, like we selected breast cancer cell as a in vitro cell culture model, then doxorubicin as anti-cancer drug model, and we performed certain cell-based assay to assess the effect of doxorubicin. That is already known. That is nothing novelty is there. But what is the novelty here? We try to explore the intracellular and extracellular profiling of breast cancer cells treated by the doxorubicin. And then we also connected in the context of mimetics. That is, we abbreviated as minas, mimetic of N1-acetyl spermidine. So I will describe that. Also, one of the technique for the profiling of intracellular and extracellular metabolite profiling, it is important to mention and describe here. We have developed an in-house uh, metabolite profiling tool that is in the form of VTGE, and also we have coined a word that is metabopsy. So today I would like to discuss metabopsy as various aspects. It can be urine metabopsy, saliva metabopsy, or nail metabopsy. So today I'd like to connect here the dots, that is cellular metabopsy. Within the cell and outside the cell, we perform the metabolite profiling. And that is the whole flow model of the VTG tool. That is, in a very simple way, it traps the biomolecules here in the matrix, and the small metabolites are eluted at the bottom of uh, the VTG tool system. And this is directly uh, uh, can be sent for various analytical techniques, including mass spectrometry. So here is the data that connects the story here. So this is one of the limited study where the intracellular the evidence on the one of the form of the doxorubicin, because the cell was treated by the doxorubicin, and also the acetylated polyamines, this one. And that is what the novelty, or you can say the incremental evidence is beyond what is known in the field of cancer biology. So here is the, if you look at here, so DMSO and the doxorubicin, obviously doxorubicin will show the highly appreciable, cell death and loss of viability. That is not surprising. Okay? Now, if you look at here, the same cells, also as a part of additional evidence is submitted for apoptotic cell death assay, and as per the expectations, more than 80% of the cells are, if you look at, showing the apoptotic cell death, and that is also expected. What is the whole the incremental data that is in the form of that the doxorubicin, one of the form of the dox, and the oncometabolites, that is the acetylated polyamines, we detected inside the intracellular compartment of the cell. However, method we have devised at our end for to distinguish intra and extracellular metabolite profiling. If you look at here, we have very clear evidence on the N1-acetyl spermidine, that is one of the form of acetylated polyamine. In case of doxorubicin-treated intracellular breast cancer cell compartment, it is important to mention here that the at basal level, like if the cancer cells are growing in a culture flash, at basal level, if no drug is given, the detection of polyamines or their acetylated polyamines like N1-acetylspermidine is highly limited. There is a very, very less data that has shown the abundance of these polyamines at the basal level. And that is why in our case also, we did not find the evidence of N1-acetylspermidine or polyamines at the basal level. That may be due to the fact that polyamines is known to work at very low concentration for the metabolic reprogram. Also, we found that doxorubicin, that is also one of the extracted ion chromatogram of the intracellular compartment of the breast cancer cell created by the dox. Okay, and that gives very clear idea that yes, truly the doxorubicin, once it is inside the cancer cells, and those cancer cells definitely mostly are dead, more than 80%. So there is a metabolic adaptations may be going on, and that might have induced the level of N1-acetylspermidine compared to the DMSO or the control. Okay. All right. So that was the whole the cell-based data we had, and then we asked question why there is a relevance of is there any relevance of N1 acetyl spermidine in the context of doxorubicin mediated cell death or doxorubicin mediated drug resistance? In other way, we thought, okay, so whether N1 acetylspermidine are able to interact with certain target intracellular proteins, right? So the intracellular protein mostly we targeted the uh, deacetylases like SDSC1, 2, 3, SDSC10, etc. So we did some basic molecular docking for the screening whether the relevance of N1 acetyl spermidine or any other acetylate polyamine as a part of target substrate or the inhibitor. Now, if you look at here, the initial data, what we found that among the many form of polyamines or their acetylated polyamines, N1 acetyl spermidine was quite better in terms of binding to the one of the target epigenetic enzyme that is SDSC10. We know that SDSC10 is involved in one of the pathway where the metabolic pathway and the epigenetics are going to be launched. Okay, so we got the basic idea that probably N1 acetylspermidine has some specific binding or interactions with the SDSC10. And then we went ahead with the idea of whether can we create some mimetic of N1 acetylspermidine based on the cell-based data and the molecular docking screening data. Can we create mimetic? And that is why that gave the idea of creation of MINAS, mimetic of N1 acetylspermidine. And I would like to display some of the uh, preliminary data. And data, if you look at here, this N1 acetylspermidine, this is the natural oncometabolites, right? This one. And this is the mimetic. So if you look at here, the binding energy is quite significantly enhanced. Definitely what our intent was, can we design certain mimetic of N1 acetylspermidine so that these mimetic can work as an uh, inhibitor of SDSC10. And then SDSC10, we know it is very well-known oncoprotein that supports the cancer or tumor metabolic reprogramming during the drug resistance. Also, it is important to mention here, this MINAS was compared with the known inhibitor of SDSC10, that is tuvastatin A. And if you look at here, the comparison of the binding affinity is almost quite comparable and appreciable. It is also important to mention that even in terms of quantitative binding parameters like hydrogen bonds or number of hydrogen bonds or their distance, etc., is quite better or even comparable with the known inhibitor of SDSC10. So it gives us also very encouraging views that potentially what we design MINAS as an inhibitor of SDSC10, it may work to an extent, right? By extending the same observation, uh, we also characterize in detail, like the visualization of the MINAS and SDSC10 complex. And if you look at here, it's very clear that this is the MINAS and this is the known inhibitor tuvastatin A, binding to almost the same pocket, and also the interacting amino acid residues, and most importantly, like SER141 and tyrosine 1 and 3, that is known at the inhibitory binding domain of SDSC10. So it gives us also very promising. Then we thought, okay, let us go for the stability of the complex of MINAS and the SDSC10. And then here is the data on the RMSD plot, that is through the molecular dynamics and simulation. If you look at here, this is the MINAS and this is the tuvastatin. So almost both non-inhibitor and the MINAS is binding, and their stability or the attributes of the stability is almost identical. And the RMSD values are also within the appreciable limit, that is below 1 to 3 angstrom. Okay. Then we also collected the data on the RMS value, that is the fluctuations of the ligand protein complex. And if you look at here, this is the domain, this one, where the inhibitory pocket is there. So if you look at here, the fluctuations of RMSA values are almost highly within the range or within the appreciable range. And that is almost comparable with the known inhibitor tuvastatin A. Okay. And then also, we collected the data on the protein ligand contact maps. If you look at the ligand protein contact map, almost it shares certain notable amino acid residues. Those are contributing to the inhibitory domain of SDSC10. So all together, we collected the data. However, the missing part is here. So this MINAS, what we designed as a potential inhibitor of SDC10, so we collected the data up to the molecular dynamics and simulation. However, their relevance at the cellular level and at the in vitro experiment is expected, and we are right now conducting those in vitro cell experiment to assess the efficacy of MINAS as an inhibitor of SDSC10 at both protein and cellular level. Also, it is important to mention that we also collected the data on the ADME toxicity profile, right? So if you look at it, very interesting to note it down, the tuvastatin A, that is a known inhibitor of SDSC10, it shows to an extent the drug-induced liver injury, but the MINAS, what we have designed as an inhibitor of SDSC10, it is almost no. And also the other attributes like AMS test or MMP, or you can say cytotoxicity is always appreciable and within the limit. And it is also important to mention the MRTD. MRTD is a bit better than the tuvastatin. So altogether, it provides us scope to think, to propose whether the MINAS could be explored as one of the mimetic inhibitor of SDSC10 in comparison to the already existing certain inhibitor of SDSC. And however, as a part of summary or the future perspective, this is one of the limited study where the acetylated polyamines are detected intracellularly in response to the doxorubicin treatment to the breast cancer cells. Also, this is one of the limited study where the mimetic of N1-acetylspermidines are explored as an inhibitor of SDSC10 and definitely as a part of future cancer therapies. Definitely the acetylated polyamines, the method for detection, method for assessment can be extended as a part of diagnostic and biomarkers for the breast cancer patients. And in future, what we propose that MINAS as a small molecule inhibitor of SDSC10 can be a part of combinatorial anti-cancer approaches, along with the existing chemotherapy drugs. And here is the future model, okay? So if you look at here, this DOX is going to get inside, and that is, this pathway is already known. The DOX is going to act upon or binding to the topoisomerase 2 enzyme, that is known. But here is the pathway. So what is the whole proposition is that due to the DOX effect or DOX abundance inside the intracellular compartment of the cell, there is also the involvement of SAT1, that is a known tumor suppressor. And this SAT1 down regulation that links the polyamine pathway with the topoisomerase mediated cell death in breast cancer cell. So here is the possibilities that, so in case of doxorubicin treatment, the low intracellular level of N1-acetylspermidine is favorable to the breast cancer cell. However, the time doxorubicin is getting treatment upon the breast cancer cell, there is a metabolic reprogramming, okay? And due to the metabolic reprogramming, the possibilities that these N1-acetylspermidine is getting deacetylated to the spermidine. So the deacetylation by the SDAC10 is the key feature of metabolic reprogramming. So what we propose that mimetic of N1 acetyl spermidine as minas can block this SDAC10 oncoprotein. The time doxorubicin is getting entered inside the breast cancer cell. So this pathway will be combined with the doxorubicin mediated cell death. So altogether, the minas as an inhibitor can work as a combination or combinatorial small molecule inhibitor along with the doxorubicin to achieve enhanced cell death. Correct, no? Definitely. This will be achieved by the altered polyamine metabolism. Correct, no? In this presentation or in this part of research work, we have shown that N1 acetyl spermidine is elevated in case of doxorubicin created breast cancer cell at intracellular. Yeah. And that is the whole story. Thank you very much to all of you. So here is the collaborators and the student those have contributed for this particular work. Thank you very much.